There's high levels of creatinine, or high levels of ferritin.
But what there's no mention of is that paper was retracted by the editors the following year.
It's type AB blood, which is Semitic blood. And so this is human blood. It's male blood. It's not blood of an animal.
The claim that the blood on this route has been tested and scientifically demonstrated to be male, I would say that there is no solid scientific evidence for that.
Immunologist who I just spoke with last week at this conference, Kelly Kearse, it's human blood, it's type AB blood, it's Semitic blood.
I've never met him. I've never talked about the various characteristics of the blood. I would have a different message.
All right. So you recently wrote an article titled Blood Transfer to the Shroud of Turin, the Washing Hypothesis Revisited. And you published this in July, so it was a couple months ago. News articles have posted this. It's kind of, it's gone mainstream. The New York Post, The Sun, MSN, Daily Mail, and Yahoo posted about it. Could you just tell us about this paper and why has this gone mainstream?
Okay, I'm not, exactly sure about the mainstream part, to be honest with you. There was a Shroud conference around the same time. So I think that was part of it that sort of gave it traction. That, and also there was another paper that came out that said that the Shroud image was more like a bas-relief than wrapping a body. So, which was sort of, I guess maybe you would call it anti-authentic. So I think the media sort of wanted to play the two off of each other. I think that also gave it some traction. But I was kind of surprised. I didn't even really know about the news articles till my brother-in-law sent me a text, and my nephew started sending me text. And they live up in New Jersey, so they saw it in the Post, and I was very surprised about it. Yeah, that it took off like that. And then there was also a series of interviews with a Shroud expert on the Tucker Carlson show, Piers Morgan show. So I think that Shroud was in the news at a lot of the time then, and also with the conference. So I think just together, that's sort of why this particular one took off.
And this paper is if the body was washed or not.
Yes.
Tell us what that paper is about.
Okay, so I'm gonna, if it's okay, I'm gonna pop up and just show some slides and kind of give just a quick background on it, and then I will talk about that. So to look at a particular idea that's been controversial and talked a lot about in the Shroud world is whether the body was washed or not before being wrapped in the shroud. So I'm going to give just a little bit of background on blood transfer, and then I'll get into the latest paper. So here we have the body and the part of the shroud that's touching the body. We'll call that the front side. And here's the opposite on the other, we'll call that the reverse side. So if you look on the left hand, this is the front side. This is what we're typically used to seeing. So here's the blood stains coming down the arm just to kind of orient things. And here's the side wound. And over here on the right, this is the reverse side, which we normally don't see that often. So you can see the blood stains coming down the arms here. And here's the side wound. So many blood stains soaked through to the reverse side. Well, how was the blood transferred? So there's several schools of thought on this. One is the blood was transferred from the body while it was wet or in a semi-liquid state. And I'll admit that that's the one that I particularly kind of favor. But others have made the point, well, Christ was on the cross for quite a while before they took the body down. Some of those blood stains would have dried. So how could you have dry blood stains transfer from a body to a cloth? Well, if you look at what the Gospels say about where Christ was buried, he was buried in a cave tomb environment. And we don't know exactly which particular cave. A lot of Catholics believe it's the Church of the Holy Sepulchre is where it was. And one thing all caves have in common worldwide is they have a low temperature and high humidity. And measurements had been made in that actual location in the Church of the Holy Sepulchre, and they fit within these particular parameters that are indicative of a cave environment. So I wanted to look at this experimentally. So I took blood and added it to skin, and I used pig skin for these experiments because that's as close to human skin as you can get. It's what medical students use to practice sutures when they're first learning how to do that. So I put the blood onto the skin. I allowed it to dry really well for about 4 to 5 hours. And just to make sure it was in fact dry, I took a piece of material and did a test blot just to show that nothing really transferred. And if that was okay, then I overlaid it with a piece of new material, and I put this inside of a humidity chamber. So what we're looking, and I pulled the material off the following day, so it went overnight. So on the left-hand side, this is a low temperature, and on the right-hand side is room temperature. I did room temperature just to sort of simulate maybe going toward the opening or the mouth of a cave. And you can see with low humidity conditions, you essentially don't get any transfer at all. You only see transfer under the high humidity conditions. This shows the contact side. So the contact side, you get nice transfer. If you look at the reverse side, you can see very similar to the shroud, these bloodstains soak all the way through. And remember, these were dry before they went encountered those particular conditions of high humidity. So here's a group of bloodstains. This is the original on the skin. And this is the control blot just to show that it's dry. And this is what the resulting imprint is the following day. So you can see that it pretty faithfully matches the original pattern. Also, the edges are relatively smooth and neat. All right, so what I've done here, I've tried to kind of mimic some of the bloodstains on the shroud. So here's the epsilon wound on the forehead. Here's the original on the skin. This is the control transfer. And here's the blood imprint the following day. Now, I got a little bit of cracking on these. I think it's probably because I sort of bent them in half pretty well when I went to take them off. But you can see, just like before, it matches the original pattern pretty well. So this brings us to the arm and the wrist region. So I chose this particular blood stain on the arm because it's been suggested that that one would have been dried. So here's the imprint. You can see it's relatively pristine. The edges are quite neat. This wrist wound is particularly interesting. This is a ultraviolet photo that was taken during the STIRP investigation back in the late 70s. So this was work done by Miller and Pellicore. They used ultraviolet light, and they found that many of these bloodstains had this fluorescent edge or halo around them. And this was subsequently shown to be blood serum. So I mimicked that one. This is the original on skin. This is under visible light, so you really can't see that serum very well. But if you look at the resulting blood imprint the following day, you can see that both the blood and the serum have transferred. And it's a pretty neat transfer. Everything kind of stays in their own lane. So this humidity imprinting, it demonstrates that dry blood stains are effectively remoistened and transferred to the cloth. Now, Barbet, who wrote the book, a doctor at Calvary, he had suggested a similar kind of idea that this happened because of the cave tomb environment, but he thought it was more that the moisture was coming from the body. These results show it's probably more environmentally driven. Now, some have made have raised the point that, well, the body would have been covered with a lot of blood, so it probably couldn't have happened this way, because if it did, there would have been a lot of blood on the cloth, which I think is a very fair point. I'm just kind of putting this out there to say this is what the results show, that maybe some blood stains could have transferred that were originally dry, and they're capable of transferring in this environment. So that's going to bring us to the question, well, was the body washed prior to placing it in the shroud? So I'm just going to show just a little bit of a background on blood clotting, and then we'll get into the new paper. So in the late 1990s, there was a series of studies by Dr. Gilbert Lavoie that were kind of classic shroud transfer studies. He put blood down onto saran wrap, and then after a period of time, he would put material on that and see what transferred. So I wanted to extend these studies in two important ways. Number one, I wanted to bring in the UV and to see what was going on with the serum. And number two, probably most importantly, I wanted to compare this with what it looked like if you added blood directly to the cloth, let's say like how a forger might have done it. So here is blood that's transferred to the cloth. This is linen. This is looking at under UV. You can see that when you use blood that's clotting, you get these fluorescent halos or edges, similar to what was described on the shroud. When you add the blood directly, there's no halo that's seen. I've tried a lot of different ways to add the blood directly, so I've dropped it on. This is using filter paper. Dropped it on. I've used a piece of wood, a piece of metal. I used cloth to transfer it. Did not see a halo using any of those. Only when clotted blood was involved in the transfer. What I've done here at the top, I let the blood clot first, and then I stirred it really, really fast and really, really hard to break the clot up. I transferred that. I didn't see the halo. Underneath, I let the blood dry really, really well for several days. I then took a mortar and pestle. I ground the blood up. I added water to that and transferred it to the cloth. Didn't see the halo. So the only time you see the halo is when clotted blood is used and not when the blood is added directly. However, I did go on to show there are ways that you can get these serum halos without clotted blood being involved. So one of these, you can use additives such as juice from fruits. So the reason I chose this, it's been known for quite a while, a number of years, that if you add lemon juice to blood, it will slow down or prevent the coagulation. So fruit juices contain citric acid, which is why in modern day blood collection, to prevent coagulation, sodium citrate and potassium citrate are used. That's the basis for that. It just happens to turn out that fruit juices are also fluorescent. It doesn't have anything to do with the citric acid. It's because of other components. But here you can see this is a clot transfer control at the top. And here's using blood with just a little bit of these mixtures. You can see it gives a very similar appearance. So that's one way you can have these serum halo or edges present without clotting being involved. Another way is just to add blood directly, no additives or anything, just blood added to the cloth. If you do a quick, pretty gentle heating, you can also see these fluorescent edges appear. Now, I don't really think that any of these the additives or heating was involved in the halos on the shroud, because if something like this had been used, you would see it for every single blood stain that's present on the cloth, which you don't. You see it for many of them, but certainly not for all of them. So I think kind of the most logical conclusion of this were that these serum halo edges indicate that clotted blood was transferred to the cloth, which is a bit of a game changer because it indicates that a body was most likely involved in this. So that brings us to this new study. Well, was the body washed prior to placing it in the shroud? Because there's been quite a bit of debate about this over the years. One thing to consider is Jewish custom. Jewish custom says that if a person dies a violent death, the body is not to be washed. And Dr. Gil Avoit, in his second shroud book, his most recent one, he does a very good job at explaining this relative to the shroud and Jewish tradition. It's not the book that's pictured here, but again, that's a book by Dr. Gil Avoit. So if anyone's interested in looking at this relation of this to Jewish custom and the shroud, I would suggest they check out that particular book. I wanted to evaluate this kind of more from a scientific perspective. So as a reminder, serum is about 92% water. It has an extremely high water content. So any proteins that are in serum are going to dissolve very, very well in water. So what I've done here, I've taken blood, put it down onto glass, and if you look over here, this is under UV, you can see that serum halo or edge. And then I did a rainwater rinse. So I chose rainwater because I thought that was probably more similar to what they might have used at that time, as opposed to modern water with all the additives, or even distilled water, which is missing a lot of things. I used rainwater, and the way I did this, I did not touch the blood stain. I was about 6 to 8 inches away from it. And I just gave it a very quick spritz, very low volume. And you can see that after the rinse, the halo is completely wiped out. This is on skin. So here's the serum halo that you see. And this is after the rinse. The halo is wiped out. This is pure serum right here. So this is before the rinse. This is visible light on the left and then UV on the right. After the rinse, it's completely gone. That's on glass. This is on skin. So here's the serum before the rinse. This is after the rinse. It's not there. Now, some have suggested, well, maybe it's kind of a speed wash sort of affair, really, really quick wash. So what I did, I put some rainwater, just a tiny bit, spritz again on the blood stains, and then I made an imprint of that. You can see it's very, very smeared and diffuse, not like what we see on the shroud, and certainly not like what we see if you compare that with humidity imprinting, which we talked about a few minutes earlier. There, those edges are quite neat. So I think these results are consistent with the idea that the body was not washed. Well, that brings us to what we'll call the washing hypothesis. And this is the subject for the most recent paper that came out back in July, the one that got picked up in the media and so forth. And probably the biggest proponent of the washing hypothesis is Dr. Fred Zugby. So he was a coroner. He had a lot of experience with cadavers and wounds. And he showed that if you took a cadaver and with a wound and washed it, that blood would ooze out. So his idea is the washing hypothesis is what you're looking at on the shroud. That blood is there because the body was completely washed, thoroughly washed, and all those blood stains you're seeing is new blood that has oozed out from those wounds. It was known as far back as the 1800s. Observations were made that normally blood is spontaneously coagulable. So in other words, it normally clots. However, it's been noted that capillary blood of a cadaver is always fluid and incoagulable, and that blood in the veins of the limbs is more often fluid than not, is only exceptionally coagulable. And in the 1960s, it's well known that the blood in sudden death, asphyxia or shock, shows fluidity but no coagulability. Now, in Zugby's own firsthand observations that he made by washing these cadavers with wounds and looking at blood that oozes out, his own firsthand observations, he said, after postmortem blood flows from a wound, it rarely coagulates, rarely clots, but it may dry. It dries just through simple evaporation. Does this seem to me to be a little bit at odds as to what was observed on the shroud, where you have these serum halos indicative of blood clotting? So in the most recent paper, what I did, I took a blood and I put that down. So this is on glass as before. Here's the fluorescent halo. I did that under normal conditions and conditions where anticoagulant was present to prevent clotting. You can see that you don't get formation of a halo. That's on glass. This is on skin. So here's the halo under normal conditions. If you impair coagulation, you don't see the halo form. Some studies have shown that the pH of blood in cadavers drops several hours after death. So I adjusted the pH of the blood to be in those conditions. I did that with and without coagulants. So here's the control under low pH conditions. You see that halo form. Under low pH and impaired coagulation, you do not see the halo present. I also want to do some of these imprinting type studies. So I put the blood down onto skin and then made a blood imprint from that. So here is the control. This is just regular blood as it's clotting. You can see the transfer. That serum halo or edge is present. And this is when coagulation is impaired. There is no halo that's present. So I believe these results show that ineffective coagulation that's associated with the lack of formation of these serum halo rings or edges. So in my opinion, these data don't really support Zugby's idea of washing with successive oozing. I don't believe that's a major transfer mechanism for these blood stains that we're seeing on the shroud. And I didn't show it here, but I also found under those high humidity conditions, if you put fresh blood under those conditions, it's actually arrested at a very early stage of the drying process. It really never dries, and it certainly never forms a serum halo. So that's going to bring us to part 2. So I will just stop there and see if you have any questions.
Are you using the same kind of blood for each of these tests?
Yes. So it's human blood.
For the fruits you're using that to make it acidic?
So no, so the fruits for the lemon juice and lime juice and things like that, I just prepared that from fresh fruits. To make it acidic, I used HCl, hydrochloric acid, and then just adjusted the pH based on volume. And then I used a very low volume. There's an equation called Henderson-Hasselbalch that you can use to figure out how much it takes to make the pH a certain number. So I did that, and I verified it using a pH test strips to see that that was approximately the correct pH. So those were two kind of unrelated things. The fruit juice, it is acidic. Those are weak acids that are in the fruit juice. And I use such a low volume. You don't need very much to get those rings. I use such a low volume there. I doubt it probably adjusted the pH of the blood that much. But those were kind of two completely different studies.
Is the blood you have still like a reddish color?
Yeah, so it's fresh blood, yes. And even overnight, it'll turn a little bit darker, as blood typically does. But you're talking, 12, 14-hour a period, something like that. Over time, that redness will not stay.
Okay. Because I know some people, that's their argument that the shroud, the blood's red.
Yes.
How is it still red after, thousands of years?
Yeah, it's a great question. I think it's easier to answer why it's not red. So looking at some of those hypotheses, I've checked out a few of them. And so, for example, one suggestion was bilirubin. High levels of bilirubin cause it to be red. So I added in a huge dose of bilirubin. I did a complete dose response from a little bit to a little bit more, all the way up to these pretty high amounts, and it did not stay red over time. I also had a blood from a genetic a rat that is genetically predisposed to high bilirubin, and I looked at that blood over time. It did not stay red. Now, there have been some results. This is from Paulo de Lazaro, who I have mad respect for all of his work. But he took bilirubin, added it to blood, then hit it with a laser, so hit it with UV, and then follow that over time. And he shows using what are RGB values, red, green, and blue, that you break down. You can plot those and see that the red is more maintained. But by the eye, just looking at it, I don't think you can really tell the difference. And these observations that people have made, they've made this for hundreds of years, that the blood is a lot redder than it should be. You have to have something, I think, that maintains the red color by eye. And I can understand, he will always say, well, if it's under a certain light, the color appearance is going to change, which I appreciate that, but I think you just bring the controls under the exact same conditions. I've gotten blood to stay red for up to about a couple of weeks. I So I was testing, Ray Rogers had a hypothesis that saponin, which is, he thought ancient linen was treated with saponin, that's what really caused it to stay red. So I tried a lot of different types of saponin and never got it to change color except for one. And the one that I did, it happened to be stored in glycerol. So when I took glycerol by itself and did it, it stayed red, but only for about 2 weeks. So it really didn't have anything to do with the saponin. It was just the carrier that it was in. But that's as long as I've gotten it to stay. So I have some other ideas as to why it's red, stuff I'm working on. I don't really want to say too much about that right now, but I think it's a very interesting quality of the shroud. It's one of those things when you first look at it that really stands out. And when the STIRP team was investigating it, that's one thing that stood out to a lot of them. It's very striking, the red color.
When I was looking online, a lot of people say that the sciencepublishinggroup.com, they say it's not a reputable like academic publisher. What would you say to that? And can you get this paper in like another
Yes,
publisher?
what I would say to that is I look intentionally. So I am a retired researcher. I have like 40 something years of experience. If someone wants to see my credentials or competence, the best thing they can do, go on to PubMed, PUBMED, and search Kearse, my last name, space KP. And that will pull up everything I've done before I was involved with shroud studies. And publishing has changed so much in the last over the 40 or so years that I've been involved with it. So there used to not be all of these huge amounts of journals online, et cetera. But now you have journals that are just junk. You can write a check, pay for play. They'll take your paper. There are lists of predatory journals. I always check out to see if anything I'm going to send to is on that list of predatory journals or not. And I can guarantee you at the time I send anything in that I would never send it to one of those intentionally. And I know some of these, they're not the maybe best mainstream, high profile type journals. But I can guarantee you every single paper I publish, I have gotten a peer review back from it. And they read and understood what I have done. So that's how I would reply to that. And I think that, as I mentioned, I think over the past 40 years, the publishing game, it's changed so much. And you will find, even just because something is in a peer-reviewed journal doesn't mean it's great work. And even because something's in a journal that may not be one of the top 10 or 20% doesn't mean that it's not good work. You kind of have to take your chances these days when you read things. So I think in the end, the truth comes out. If it's good work, it will stay and it will be reproducible. Whether it's published in a flagship journal or one that's more lower tier. So I'm fully aware that a lot of these things aren't in the top journal. I try to put them in as high as high as I can.
Did you want to get into part two?
I will get into part two. So what part two is, we're going to kind of switch, and I'm calling this. It's about the bloodstains. I'm calling this a scientific fact check, just to sort of see here in 2025 characteristics that have been described for the blood on the shroud, how scientifically accurate those are. So I've kind of come up with these three, I call them maxims that I want to just present first. These are kind of ground rules that I think need to be followed. So objective and unbiased presentation of scientific information is essential, even when the results may be inconclusive. A desire to build a case for shroud authenticity, this should not necessitate the need for embellishment of scientific results. Number three, exaggerations about the thoroughness and validity of any shroud blood studies. This only serves to erode any previous credibility that may have been legitimately established. So I think those are the ground rules that have to be followed when any kind of presentation about shroud characteristics. Now, today we're talking about blood's characteristics, but really any characteristics in general. So I want to play a clip from this recent interview. This got a lot of press. This was an interview on the Tucker Carlson show. So we'll just listen to what's said here.
So, okay, but of the hundreds or thousands of shrouds like this that exist, why do we think this one has an image of Jesus on it?
Because all of it matches the way in which Jesus was crucified. And that's what's powerful about the shroud.
Okay. For example?
For example, on the shroud, we have blood all over it. And the blood is interesting. It's been tested. It's type AB blood, which is Semitic blood. The fewest amount of people in the world, only 6% of the world's population has type AB blood. And so this is human blood. It's male blood. It's not blood of an animal. It's not a hoax. You would have to actually kill someone if you were trying to reproduce the shroud, because we have premortem and postmortem blood all over the shroud. So that's interesting. So this tells us that someone died.
Yeah, let's see. Okay. So I want to just say at the start, and I'm not really trying to pick on this one particular interview or this one particular shroud expert, because these kind of presentations, you can see them all over YouTube, where they're made in churches and auditoriums, and they'll repeat these same kind of things. As blood has been tested, it's been scientifically demonstrated to be male, human, AB, and so forth. You'll also see a lot of these claims on many websites that say verified facts about the shroud, verified scientific facts about the shroud, blood characteristics. So I just want to go through each of these characteristics one at a time, talk about what data actually exists and what an interpretation of that would be from my viewpoint. This is a viewpoint of a immunologist. So the first claim is the blood on the shroud is male. It's been tested and it's been shown to be male. So I'm going to give a really quick background on the particular technique that's used for this, because I think that's helpful in understanding it. So the technique is called polymerase chain reaction, or PCR. This shows nuclear DNA. This is all the nuclear genome, genes that would be needed to encode a human being. There's about 25,000 total genes. What PCR does, you make a probe to one of these specific genes that you're interested in. It will go in and find that one particular gene among these 25,000 or so total genes. After locating it, it will copy or amplify it in a chain reaction fashion. So that's why it's called the polymerase chain reaction. And it's so efficient at copying, it can make about 1 billion copies in around 3 hours or so. So if you have a situation where there's not a lot of DNA, through PCR, you can amplify it up so that you have enough to study. So here's a real life example of that. This is a study that we did last year. So this is DNA from wheat wafers. These were baked at a pretty high temperature. So that's not really DNA friendly. You expect the DNA to be very degraded and to be very fragmented. And when we run this out, that's exactly what you see. Now, if we take the same material and through PCR, we look for one particular gene. Here you can see that we have a signal. We expect it to be the size of where the arrow is, and that's what we got. And then what we do is we take that material and we sequence it just to verify that that really is that particular gene that we're looking for. So even in cases when the DNA is in very bad shape, degraded and fragmented, a lot of times using PCR, you can get enough of a signal to look at. So as far as the shroud goes, studies that were done to demonstrate that the blood on the shroud is male, there were 2 of these that came out in the 90s. And none of these has ever been published in a peer-reviewed journal. One was in a book. One was in a Shroud magazine, Shroud Specialty magazine. So probably the most famous is the one in the book, The DNA of God by Garza Valdez. So what he did, he took bloodstain fibers. He did PCR for 3 genes. He did the X and the Y for sex determination, and then also did a gene of hemoglobin. It's called the beta globin gene. He got a positive signal for all 3. One of the problems you always run into with any type of DNA study is contamination. So here's a photo. This is back from the STIRP investigation in the late 70s. 50 plus scientists examined the cloth for 5 days straight. Here you can see they have their hands all over it, and none of them were wearing gloves at the time or masks or any kind of protective equipment, because back then they didn't really know. DNA science hadn't really been developed. Here's another picture. You can see they grabbed it, have their hands on it. This is an investigator. He's actually right on top of a blood stain. So he's laying on it. He's also got another instrument in his mouth at the ready here, no mask, etc. This is from Garza Valdez's book. This isn't actually Garza Valdez, but this is the person that's sending him the samples. You can see there's no gloves. You can kind of make your own mind up as to whether you think he's really wearing the mask or not. But contamination, it's always an issue in any kind of DNA studies. So Garza Valdez, the way he addresses the issue of contamination, he says, because we got a positive signal for this beta globin gene, this rules out contamination. Unfortunately, what he overlooked is that skin cells also express the beta globin gene. I think there was some confusion there between protein and DNA. The DNA of most cell types is the same, and skin cells certainly have the beta globin gene. So this in no way rules out contamination. It in no way shows you that this signal comes from blood cells. Again, this is in a book. You can write whatever you want to and publish in a book. The second study that was done, this is in the Shroud magazine. There they reported that both male and female DNA was found, and they were very upfront about it. When they look for polymorphic indicators, what those are just markers that are unique to individuals. So if you want to see how many people you can account for, how many profiles are present, they found in those samples that they could account for at least 6 different individual profiles. So clearly there's contamination there present also. So that is it as far as scientific evidence that the blood on the shroud is male. So I've written an article about this. Here's the reference if anyone wants to go and look that up. And let me just say one more thing about DNA, and then I'll just pause this for questions. So anytime you're trying to identify that a DNA signal comes from blood, it's always somewhat difficult because of the problem of contamination for any kind of DNA study that you're doing like this. There's no studies that currently exist that demonstrate that a DNA signal has been obtained from blood cells on the shroud. You have to validate that a DNA signal truly comes from blood cells. To do this, you would have to use unique gene segments that are blood specific. And this is difficult because, as we said a few minutes ago, for most cell types, the DNA is exactly the same. Now, from an immunology perspective, there is a way that you could do this if you analyzed immune receptor genes. Immune receptor genes are found in skin cells as well as blood cells, particularly the type of blood cell we're going to talk about here is a lymphocyte. And what's unique about the lymphocytes, they have the genes just like the skin cell. At the genetic level, the DNA is identical. But what happens with the blood cells, the DNA undergoes a cutting, pasting, rearrangement as part of their normal maturation and development. So it doesn't stay in this original configuration. It's literally cut and pasted together into a unique rearranged form. Blood cells are the only cell types in the body that do this. So if you use PCR, you can make a probe that's specific for this rearranged form of the gene. And if you got a signal, it would be guaranteed that it's coming from blood cells. In this case, it would not matter how many thousands or hundreds of thousands of people had handled the shroud throughout its history. That signal is unique to blood cells. Now, you couldn't use this to determine whether it was male blood or something like that, but you could, if you were able to get a signal, you could potentially sequence this and look at the species of origin, human blood, which we're going to talk about in just a minute. So I've also, that's also mentioned in this same article if someone wants to check that out. So I'm going to pause that here. And Joe, if you have any questions about that, I'll be glad to answer them. So the summary of it, I would say the claim that the blood on the shroud has been tested and scientifically demonstrated to be male, I would say that there is no solid scientific evidence for that. But it's repeated, in this presentation, it was repeated. It's repeated in many presentations. There's a lot of this has what I call like an echo chamber effect. One person repeats it, the next, so forth, so on, so on. But if you go back to the original studies and look at them, this is the state of the art. Scientifically, this is what we have.
I was just curious, how long does a skin cell last?
A skin cell, it's not going to last. It's not going to last probably more than a few days or so. It's going to be dehydrated and start to be degraded. Now, some products from that may still kind of hang around, but it's going to be chewed on by bacteria. But some part of that could potentially survive. DNA, it's a lot to ask. It's a long time and environmental conditions, but maybe a possibility that it might be. I mean, they've looked at DNA under people that do that under very rough conditions and with very, very small amounts of DNA. Those that are experienced in that type of analysis, it pulls some pretty amazing things off. But yeah, time is definitely going to be against you in that.
Because I remember you talking about that, and you were saying like just one person sheds over 100,000 skin cells a day.
It's incredible. I've even seen it in the millions of range. And that outer layer doesn't really have, those cells are anucleate. They don't have a nucleus, won't have DNA, but the layers underneath do. And so this STIRP team being there in the 70s and more recently, the samples taken in the 90s and things like talked about in the Garza Valdez book, a really good chance that DNA is still around on there for sure. So that's not that long of a time in the DNA world of things.
Do you think the new pope will let anyone test the shroud?
I honestly have no idea. I think that remains to be determined. Now, in this Tucker Carlson presentation, the shroud expert there. Ooh, were we going away, Joe? Are we back? We okay? My thing just went blank. We okay?
Yeah, we're back now.
Okay. All right. He made statements like the Catholic Church is afraid of the shroud. The Catholic Church is indifferent to the shroud. The Catholic Church is not interested in educating people about the shroud. You can watch the full-length interview and you'll see those statements. I thought they were, I'm a Catholic. I was pretty offended by it, to be honest, as a lot of my Catholic friends were too. I think, the Catholic Church, they've never taken an official position that it's authentic or not authentic. I think it's because they're wise and they're patient. They're neutral on it. So I think those statements I thought they were very unprofessional to begin. But you can watch that full-length interview. You'll see them. There's also some remark about I have an allergic reaction to Catholic relics, things like that. Very non-professional, my opinion. Offensive to Catholics. So I don't really know. If the Pope would allow future testing. That remains to be determined, I would say. Yeah.
Do you think it'll be like on display for people to look at?
Don't know. That's another good question. It usually comes around every 20 or 25 years or something like that. So I don't know if they would increase that timeline or just kind of keep it to where it is. Yeah, good question. I feel fortunate. I was able to see it I think it was 2015. So I was able to get in on that, but I don't know when the next exposition don't really know.
When you saw it in person, what was that experience like?
It was incredible. And it was really cool. So the way it works, it's free to go in, but you have to have a ticket. So it's like a timed entry kind of thing. Okay. So I went with a group. It was actually the group I went with was led by John Jackson, who headed the STRP team. So you go in and you stand. I was pretty surprised how long we were able to be there, maybe 5 minutes to go in. It wasn't super crowded when, excuse me, when I went in, but you stand there and look at it and you're about, 12 or 15 feet back or something like that. And, there it is. I mean, the thing you've seen so many pictures of, and there it is. And so after you do that, you can go to the back of the church and there's a sitting area back there, kind of a little, because it's in a church, sort of a prayer type area. And there you can sit as long as you want and look at it from a distance. So I did that. I actually went through, I think it was 4 or 5 times. So I got tickets to like 4 or 5 times, and I went back several cycles. I went over there once just with John Jackson, just the two of us, which was kind of cool because he was sort of telling me about the study and where they brought it out and things like that. So I tried to, I tried to look at it in different ways, sort of scientifically, sort of, okay, I'm going to meditate faith. I'm just going through here and do a faith kind of meditation on this round. So I tried to look at it as from different perspectives, but it was absolutely awesome being able to see it in person. Yeah, it was incredible.
Did anything like change your mind on it after you looked at it in person, or
No,
no?
no. So I'm one of those that I don't consider myself pro-authentic nor a skeptic. I just don't think enough work has been done. So I am honestly unsure for me whether it's real or not. And I was able to take some photographs of it. So, if you're kind of discreet and respectful, I had just a point and shoot when I was up close, but then I had one with a bit of a zoom on it when I was in the back. So I took some photographs of it. I was surprised how red the blood was in person, so I was kind of keyed into that. One thing I was going to look at, it looks just like the pictures and things that you see, but in person, I was sort of surprised how red it looked. And I was able to, a few years back, Dr. Wanger had samples that were taken from the Shroud. He was gracious enough to let me look at some of the blood samples for an afternoon in the microscope, and I was surprised there how red the samples were, even on a microscopic level. But yeah, that's one thing that stood out to me in person when I went through. Yeah, it was a fantastic experience.
I'm surprised they let you take a picture. That's pretty cool.
Yeah, they would kind of. It would sort of depend on who was there, and I guess maybe how big the camera. Because I would kind of hear no, to some of the people that were doing it. Flash was off the table, but yeah, I just had a little guy and just kind of stood there, and they were okay even in the back, but kind of discreet, respectful. It was yeah, they were okay with it. Yeah.
Is that stored in like a temperature controlled?
Yeah, it's like temperature controlled. It's stored in essentially argon, primarily argon in a glass case. So if you go there like today, it would be in the case horizontal on a type of altar with a cloth over it, and then kind of all behind glass. So you couldn't really see the cloth itself. When it's displayed, it's sort of up like this, and then in a frame that's kind of lit up. And big, like 14 foot long. So Okay, so we're going to go to the blood on the shroud is human. So as far as the blood on the shroud being tested and showing that it was human, those were done back in the early 80s by Bima Bologna. He's an Italian scientist. And then Heller and Adler, they were associated with the STIRP team. And these were studies done using immunological methods. What that just means that antibodies were used. There were essentially two major blood proteins that were looked at, immunoglobulin and albumin. Bima Bologna kind of concentrated on immunoglobulin. Heller and Adler concentrated on albumin. They also looked at immunoglobulin a little bit. The results they were essentially the same, so I'm just going to talk about them somewhat collectively. So again, these are serological methods. They involve antibodies. So we have here a preparation of an anti-human antibody, so against human blood proteins. So antibodies themselves are very, very small. You really can't see them with the naked eye. So what's typically done is they add some kind of fluorescent or chemical tag to them. And then if you look at this under the microscope, wherever it lights up, then you know that the antibody has found its particular target. So they took unstained fibers from the shroud as a control, fibers without blood, and then bloodstained fibers, reacted to them with these anti-human antibodies that had the tag. And they found that only the bloodstained fibers lit up, the control fibers were negative. So what they concluded is that human blood components are present in the bloodstained fibers of the shroud. An immunologist is going to look at this and they're going to say, well, I think you've shown blood components are present in the bloodstained fibers, but I'm not really convinced that you've shown that human blood components are present. And just to point out, these results from either group, these were never been published in a peer-reviewed journal. But here is why an immunologist would say, well, these experiments don't really demonstrate that human blood components are present. When you have this anti-human designation, what that refers to is how the antibody was made. It really doesn't speak to how the antibody may react. And sometimes if you're not really in tune with the immunology, this can be overlooked. So what it means when it says anti-human, it just means someone took a human protein, they put it into another animal, and that animal's immune system made antibodies against it. It doesn't speak to how the antibody may react. In immunology, there's something called cross-reactivity. So you can make an antibody against a human protein, but it may in fact react with the same protein in a variety of other species. And this is exactly what the case was. So cross-reactivity, when you're trying to demonstrate species of origin, it's an absolutely critical issue. Bima Bologna never addressed this in his studies with immunoglobulin. Heller and Adler did, but it was relatively limited. So they did test several species with their antibodies. So they found it was positive with human blood, also blood from chimp, some reactivity with baboon. So that's not really that surprising. They tested against cow, I believe it was cow and horse or cow and pig. Those were negative. But what we now know, some 40 years since then, is that the cross-reactivity is much greater than previously considered. So if you expand this and you look at reactivity of those antibodies with blood from other species, we now know those antibodies would have reacted with blood from rodents, from cats, from dogs, and a variety of other species. So you can't really make that claim that it's been scientifically demonstrated that the blood has been shown to be human, that it's been tested and the test showed that it's human. This is what the International Center for Studies on the Shroud, this is located in Turin. This is the Turin Centro. This is the only center in the world to be officially recognized by the pontifical custodian of the shroud. This is what they say as far as the species of blood that's on the shroud. They say Heller and Adler did not conduct extended tests to rule out that the blood belonged to a species other than humans. You can find this on their website. They also say the identification of the species of the bloodstains on the shroud is still a subject of study. I think that's a very honest way to say it. It's where the data currently stands. It's a much different message than what we heard in the interview on the Tucker Carlson show or on numerous presentations that are made by shroud experts. You can find these on YouTube, or people that just go on and talk on YouTube. Also on many websites, they have verified scientific facts about the shroud. They'll just say the shroud has been scientifically determined to be human. Much different a message than what we see here. Now, I published a paper on this. Here's the reference for it. If anyone wants to read that, I'll just hit the highlight part. As discussed, cross-reactivity precludes a definitive assignment of human or even primate blood being present on the shroud. The blood is most correctly classified. This is from a scientific standpoint. The blood is most correctly classified as species unknown. Now, I want to talk about this. So this is new. This is something that got picked up in the media in response to some of these interviews that were done. So we're going to talk about M, N, and S proteins. These are three different proteins that are found on the surfaces of red blood cells. The S is the one that's particularly important. So here's an article that came out from one of the interviews from the Tucker Carlson show, Turin Shroud Mystery Solved by the expert who says he even knows Christ's blood type. The AB blood type was first documented in 1982 by Italian biographer Dr. Pierluigi Bologna and colleagues. We're going to talk about AB in just a second. But it says subsequent testing also detected M, N, and S antigens, confirming that the shroud was unquestionably human. So the studies that have been done on the shroud fibers for M, N, and S, again, S is the most important. There's one single study. This is not published in a peer-reviewed journal. It's only in a shroud magazine. It was a very brief article. These are immunological methods, which again means antibodies were used. He reported that this is Bologna's work, that the shroud fibers were positive for M, N, and S. Now, ironically enough, in this article, there's no results that were actually shown for the anti-S reactivity of shroud fibers. There's only a written description of fairly good binding. Now, what fairly good binding of the antibody means, who knows? But Kearse, pointed out in about 2012 that S antigens are specific to humans. So that's potentially interesting. However, more recent data since 2012 has shown that anti-S antibody preparations, they're not suitable for species determination. And where this came about is in the field of xenotransplantation. Now, what that means is in a way to deal with the organ shortage in humans for transplants, xenotransplantation just means you take an organ from one species and transplant it to another, such as a pig organ into humans. When they started to do that, they ran into problems, and what was revealed from those studies is that human antisera, including these anti-S, would fall in that category. They contain antibodies that will react with blood from many animal species. It's because of a particular galactose residue. So such antibody preparations cannot be used for species determination. So even though they might have had the anti-S, they would have also had these other antibodies present, which would recognize blood cells from many different species. There are modern antibody preparations that exist that will only recognize human blood cells, but these to date, they've not been utilized in shroud research. So I've written about this. This is particularly about the MN and S. The S is the most important. Such antisera were particularly prone to cross-reactivity with blood from a bountiful number of other species. These findings underscore the necessity of using more current serological tools. That just means more current antibodies in any future investigation of bloodstain artifacts such as the Shroud, particularly in relation to species determination. Now, I will point out in fairness that I believe there is indirect evidence that exists, such as blood flow patterns, particularly the work of Gil Lavoie, and these serum edges or halos, some of the things that I've done. This indirect evidence, it suggests that blood was transferred from a human body, but that evidence is indirect. There's not a direct scientific demonstration that exists that the blood is of human origin. And I think it's said in these interviews and presentation, well, the blood's been tested, it's been shown to be human. That implies that these direct type of tests have been done and that the results were very clear. Again, this is what the International Center for the Studies on the Shroud, the Turin Centro, the identification of the species of bloodstains on the Shroud is still a subject of study. Okay, the blood on the Shroud is type AB. So studies from this is also from Byman Baloney's work. This was done in the 80s, never published in a peer-reviewed journal, just in a Shroud magazine. These were studies using immunological methods, so using antibodies. This is a pie chart that shows typical blood type distribution in the population. Most people are type O. In a close second place is type A, and then you kind of have a pretty distant third is type B, and then the rarest of all is type AB. And there's some variation depending on geographical region, could be like 5 to 8%, but AB is definitely the rarest blood type. The Shroud of Turin reports this blood type, a suggested head cloth that was wrapped around Jesus. The Siderium also typed as AB, a proposed robe that Christ wore on the way to Calvary, types as AB. Every Eucharistic miracle that's been scientifically evaluated for this to date also types as AB. So it's been said it's a statistical impossibility that this could happen by chance. The odds are greater than, say, 3.2 million to 1. Some even put this in the billion to 1 range. A recent article, Catholic Answers magazine. This is mathematical proof of the real presence, proof that God is real, proof that our Lord has AB blood. Well, if you look at what the structures are, the AB antigens, so they're composed of carbohydrates, or in other words, sugars. And this middle part of A and B is exactly the same. What makes it unique is this particular sugar side chain that's there. So these are composed of sugars, carbohydrates, which are used in a variety of organisms. In fact, many common bacteria express identical A and B antigens on their surfaces. So it's not only expressed on human red blood cells, but also on many common bacteria. And many of these objects, significant bacterial and fungal contamination has been reported. So I think it's something that you have to take into consideration from a scientific standpoint, that it's known that many common bacteria share these antigens, which could provide an explanation for these results, why everything keeps turning up as AB. These types of studies that were done, they were serological methods using antibodies. Antibodies cannot distinguish between bacterial AB and human AB because the structure is 100% identical. There are ways you could sort this out at the DNA level to really see, well, is this signal coming from bacteria or is it coming from human? But those kind of things have not been done. I want to point out, I'm not the only person that raised this question. Alan Adler raised this years ago. He was the blood chemist associated with STIRP. He seriously questioned the AB findings because of this very issue, that such molecules are shared with other organisms. Here's a recent review. This is nothing related to the Shroud. This is just on AB typing methods in general. And all of these types of methods, including some of those that were used, you can see here it says they're very unsuitable, often inconclusive for aged dried stains because of these very issues. This is what the Turin Centro, the International Center for Studies on the Shroud, has to say. Pierluigi Bologna identified the AB blood type of the bloodstains. However, the results were affected by the possibility of cross-reactivity. A very honest way to present the scientific results. Certainly much different than this kind of slam dunk that it's been shown to be AB that's often said in that fashion. So I've written about this. Here's the references if you want to check out this. I think the AB is a difficult one for a lot of the Shroud crowd to kind of let go of because they want to use that to make a connection between these various articles. But it should be pointed out that shared AB it's not scientifically sufficient to demonstrate the same origin. You would have to use polymorphic markers. So in other words, markers that are unique to individuals, like nuclear DNA STR regions, typically done in DNA profiling, or mitochondrial HV regions, a similar kind of thing, or even HLA antigens, which you could do. Those are transplantation antigens. So if someone goes in for a transplant to see if they're a suitable donor or recipient, you could also use those as markers to evaluate this kind of thing. You'd have to use something like that to make the claims of a shared origin. I did want to give a shout out to this one particular website. It's a British website called shroudofturin.uk. Unlike many of these websites that promotes information about the Shroud, I think they have it straight. It says the blood group could be AB. Additional tests are needed before this classification can be considered to be reliable. One last thing here in the Tucker Carlson interview, it said that testing from the 1990s identified blood type AB on the Shroud. So the 1990s, Bologna, he's the only one that has done blood testing for AB on the Shroud. In the 1990s, he published a book. It's called Sindone or No in the 90s. It doesn't contain new studies. It's just talks about the previous one that he's done. So I think what happens, some people use this as a reference, then other people use it as a reference, and someone else uses it as a reference. So they just kind of look at it without really going back to the original source. So in the 90s, there was no new blood typing studies. Garza Valdez, he's the guy that did the DNA of God, the blood is from a male. He said that he did test fibers with anti-B, but he didn't use any anti-A because he didn't have it. So I don't really consider that a test for AB. Bologna is the only one that's done it. And I want to just go on record and say I have a tremendous amount of respect for Bologna's work. He's the pioneer in this stuff. He's the one that started it all. And when he took the samples, he did it directly. He just used forceps and pulled out the threads. He didn't bother with sticky tape or something like that, it might give you some problems for certain tests down the road. So he just did that directly. He's up in his late 80s now. I've communicated with him several times through emails. He was kind enough to send me a copy of his latest book. This was in 2022. Actually wrote me a nice note, autographed it for me. But he should be respected. He's the guy that really led all this off. So I think the message that the blood is tested to be AB, it's by no means a slam dunk. There's inherent problems with these type of serological tests. I'm just going to jump in real quick and talk about the second kind of follow-up on AB, and then I'll come right back. So let me just share the screen, and we'll do this, and then you can ask questions about that. Okay, so related to this is a thing that was said in the interview and that's been repeated. This particular quote's from another interview that was done, but same Shroud expert that was on the Tucker Carlson show. AB is Semitic blood. So he said that on the Tucker Carlson show. Only 6% of the world's population has type AB blood, and most of them are found in the land of Israel. Well, this is 50% accurate. Only 6% of the world population, yes. Most are found in the land of Israel. If you go to the NIH National Library of Medicine database page, they have a page about blood type. So blood type AB is the least common. It is also the rarest of the blood groups. They say this is NIH database, National Library of Medicine, 2025. It is most common in Japan, regions of China, and Koreans, being present in about 10% of these populations. So it's actually the highest in Asian locations. You can scroll down this and you'll find that they show geographical regions. The Middle East region, it's a pretty comparable to what you find with other regions. So where this might come about, this idea that it's Semitic blood, it's enhanced in the Jewish population. There was an older study done in 1977 with ancient skeletons from Israel, about 1,600 to 2,000 years old. There were about 60 or so skeletons used in the study. About 19% of them were undiagnosable to any blood type at all, and about 51% of them showed up as AB. But these were studies done using these serological antibody-based methods, which are inherently unreliable, like we previously talked about. And this is just one study. You cannot take this and extrapolate and say that it's really high, AB expression is really high within the Jewish population. Now, one thing I always do in anything related to shroud studies, human characteristic, it's male, anything related, I always solicit opinions from outside scientists where that work is their specialty. People that are not really interested or working on this route, I just ask them objectively, well, in this situation, what can you give me your perception of it? So this is a perception. I ask various geneticists and people that do population analysis and that study blood type. What about this claim that AB is Semitic blood? So a consensus reaction I got, these are professional practicing scientists. The claim that AB blood is Semitic blood is not valid. It's not true. I asked them point blank, is AB Semitic blood? The consensus was no. There's the comments, there's no way to have an idea of what the relative percentage of AB might have been 2,000 or so years ago. Dr. El Hayek, he's actually an Israeli-American geneticist and a bioinformatician, he studied at Johns Hopkins, University of Sheffield, and is currently at Lund University. One of his direct quotes is, "These are very old studies that are not done anymore." I asked him about this 1977 study. Also, this is a very old field that no one is taking seriously today. So I think by no means is it, a slam dunk that the blood type on the shroud is AB, or that AB is Semitic blood. The database, the NIH National Library of Medicine, that says otherwise.
When you do your test, what type of blood are you using?
I'm using human blood. Human blood.
Blood
Healthy
type.
volunteer. I'm sorry?
The blood type of it.
Various. I've tested people with different, including AB, but different types. Yeah.
I'm assuming you get the same result.
Different blood type, so I've honestly not really done like each particular blood type for every single kind of test I've done, but I really don't think it would make that much of a difference as far as drying and things like that.
Have you heard of that? I think they call it golden blood. It's like a super rare blood type.
So the super rarest would, so like AB negative, something like that. Is that what you're referring to, the RH factor? I haven't really heard the golden blood thing specifically. The Rh factor, so that's an additional protein that may or may not be expressed. So if someone's AB negative, they don't express it. AB positive, they do. I've asked Bima Baloney point blank, hey, did you ever do Rh testing on shroud samples? And he told me no, it has never been done. But you'll see things on the internet, well, the shroud is AB positive or the shroud is AB negative. That has never been evaluated in those samples. So it could be AB, but I don't think there's really scientific evidence to support it. Those tests, they're inherently AB, that particular blood type, especially when you do certain types of tests, that's the one where it's really inconclusive. And you have all these objects that are showing up as the same blood type. I think it could very easily be explained by just bacterial contamination, bacterial presence, which independently, that's been demonstrated to be there, as far as that shroud has been said to be pretty heavily contaminated.
And Jeremiah Johnston, the guy that went on the Tucker Carlson, he also went on the Piers Morgan show, and he mentioned your name on the show. And some people might think he was endorsing you on that.
We have type AB blood, human blood, and there's some very fascinating work from an immunologist who I just spoke with last week at this conference, Kelly Kearse. It's human blood, it's type AB blood, it's Semitic blood.
Yeah, so I watched that show, and he made a comment that there's some fascinating immunological work done by Kelly Kearse, whom I recently spoke with at the Shroud Conference, and then kind of the blood is human, AB, something right after that. Well, I have never met him. Okay, he was at the Shroud Conference. He was in the audience. He asked me one question after the talk I gave. Do you find evidence for pulmonary fluid on the shroud? That's the only interaction I've ever had with him. I've never met him. I've never talked about the various characteristics of the blood. I honestly didn't know who he was. The reason I knew it was him when he asked the question is because he gave a talk 2 places before mine in the same room. But I have never, his message is the characteristics he's saying that are so well tested and known. I would have a different message. I have never met him. There at the Shroud Conference, I guess we spoke in that I answered a question from the audience. I answered quite a few. So I don't know. Maybe that just came out. You got me.
He also said that Jesus had a heart attack. I don't know if there's evidence of that Jesus had a heart attack on the cross.
I've asked physicians about that, and the exact cause of death, I've heard different things. Yeah, so I don't really have an answer to that one. I haven't really looked at that particular, kind of any in-depth study or that. I just kind of casually read some of that and asked. I'll talk about some of the evidence for polytrauma, which it's connected to what you're talking about. That's actually going to come up next. Okay, so evidence of polytrauma, high levels of creatinine and ferritin. So creatinine comes from creatine. This is indicative of kidney failure. Excuse me, and increased ferritin, that's part of an acute response, indicates that someone has a significant amount of inflammation. So I'm going to play this clip. And the reason I'm playing these clips, I just like for people to hear it exactly how it was said, rather than me sort of trying to repeat it.
How does it kill you?
It kills you in a variety of ways. It doesn't kill you quickly. It maximizes torment while minimizing it actually maximizes the length of death, and it prolongs death. And so when we study the blood work, so there are some amazing hematological reports that I've enjoyed reading thoroughly. When we study the blood that's on the crucified man, it bears correspondence with that there's high levels of creatinine, which means he was suffering from kidney failure, high levels of ferritin. His body had inflammation all over it. He was dehydrated. You read John's Gospel. Remember in John's Gospel, Jesus.
So he makes a reference to high levels of creatinine, high levels of ferritin. This is from a study done in 2017. But what there's no mention of is that paper was retracted by the editors the following year. So I didn't put the word retracted across the paper. This is how it looks online. If you go to look this up, this is what it looks like online. They put this retracted all across the front of it. And then they also explain their reasons for the retraction in a separate article. So here's part of that. This is what the editors say. This is about that study. This is the one they're talking about. High levels of creatinine and ferritin in 2017, retracted 2018. We don't hear anything at all about a retraction. So the editors were concerned about the validity and conclusions and reproducibility of the results. Further, the editors voiced there are not sufficient controls to support conclusions referring to human blood or physical trauma. So we hear about the high levels of creatinine and ferritin in this study, 2017, retracted by the editors the following year, 2018. Now, one of the articles on the retracted paper is written another one in 2024 about Jesus's tortures. He does mention creatinine in that paper, doesn't mention ferritin, mentions creatinine. And amazing to me as a scientist, he actually references the retracted paper. So it says, this is a new figure of creatinine, recognized as creatinine, already mentioned with ferritin in a paper retracted for reasons the author disagrees with. Well, I'm sure he does, but the editors thought differently. As reported in the cited paper, this is the retracted one, this is in the 2024 study. As reported in the cited paper, the presence of numerous creatinine particles with ferritin confirms at a microscopic level the very heavy torture suffered by Jesus of the HST. HST is Holy Shroud of Turin. So we have this mention of high levels of creatinine and ferritin have been found, but zero mention that that study was retracted. Retracted in 2018, it's now 2025. So I think there's kind of a general trend in a lot of these presentations and interviews. You either don't know the work that well, or there's an intentional exaggeration to kind of steer it a certain way to build a story. And how can you talk about that that's been shown without even mentioning that it's been retracted or even talk about it at all?
Yeah, I see. I don't think the guy is like malicious or trying to. I think he's just excited about it, maybe, and he just.
In science, you have to be objective, and you don't want to. You make exaggerations. I think what happens in the long run, it ends up doing more harm than good. You would want to err on the side of caution rather than not saying a complete story. So he said, he's read things thoroughly, et cetera, et cetera. And a lot of these presentations, I don't know. I'm just showing what's there.
Is there anyone else that does blood tests today like you do?
Yes. So one of the authors on that retracted paper does, and some other people work on it. I kind of the clotting and stuff maybe do more of that, or the halos under UV have done probably most of that. But there are some that study. But as far as the immunology side of it and commenting on that, no, not really. But I, as I mentioned before, I've solicited opinions from lots of different umpteen immunologists, a lot of who I know and a lot of who I don't know, and they were 100% within agreement. Even the Turin Centro's own page uses caution in saying the blood has been shown to be human, or the blood has been shown to be AB. I think that's something I would encourage people that are interested in this to, do their homework and read about it, but also ask, professional scientist about this, well, look at this data. What do you think? And from the immunology standpoint, just mention cross-reactivity and controls and see what they think. I think that's what really needs to be done with Shroud research. I think more outside scientists should have a look at this. I mean, there are definitely, scientists that are working on it, professional scientists that are working on it. But I think the more outside sort of people you have, and to keep things objective, that's what's really needed. You cannot stretch the data beyond where the conclusions go.
Is there anyone that's done tests on the Shroud? It doesn't have to be blood, but just any other tests you think's interesting or new?
I think almost all of it. Is interesting. New right now. I think we're kind of in a bit of a lull. I don't think there's really been anything new. A lot of the Shroud conference, you kind of just hear similar sort of things repeated, or certain theories, hypotheses, but without a lot of really new evidence. For example, the image. I don't think we really know the image properties. You'll hear a lot that it's cellulose that's been undergone dehydration and oxidation. But a member of the STIRP team, Ray Rogers, he was adamant that cellulose was not involved. It was actually a layer on top of the cellulose that was really involved in image formation. So I don't think scientifically that has been sorted out, but you'll hear it talked about. It's kind of a slam dunk, a done deal. Radiation is what caused this, and it caused the cellulose to undergo these changes, which certainly could have happened. I'm not opposed to any of that, but I think you have to, more evidence is needed. Okay, so this was another thing that was said, that there's premortem and postmortem blood all over the Shroud. So let me just start this out by saying there are currently no established scientific tests that allow antemortem, that's premortem, that allow premortem and postmortem blood to be identified on a 700 to 2,000-year-old cloth. You could do tests on blood that's, hours, weeks, months, maybe even up to a year or so, and tell if it's premortem or postmortem. But there are no tests that allow you to take fibers from a cloth like this, especially one that's been exposed to various environmental conditions, and test it and show scientifically, well, that's premortem blood or that's postmortem blood. I think the way this is presented, it kind of sounds like those types of tests have been done. If you're evaluating this scientifically, you have to be able to separate the blood stains from the image. So you can't use the context of the image to objectively look at it. You have to just look at the blood stains as they are on their own. And I think personally, you even have to move this away from any narrative in the Gospels. To look at it objectively, scientifically, you just have to look at the blood stains on their own. So where this premortem and postmortem, where a lot of this is coming from, this is from the same 2024 study. This is the one with the reference to Retracted Paper on the creatinine and ferritin. So it says right away at the start, analysis was focused on 3 different types of blood substances, which via the Gospels can be correlated to different moments of the passion and death of Jesus. So I think right away, a bias has been introduced to this, but let's just keep going. So here, this is a picture of a red blood cell that is suggested to be a red blood cell, a little bit smaller than normal, but there's explanations for that. And then over here, so this is under a high-powered microscope, this is an element profile. It doesn't demonstrate that this, in fact, is blood. Any kind of organic material could give you this type of profile. And there's some elements you'd like to see in here, like a nitrogen, sulfur, that aren't. But it says here, identification with postmortem blood that came into contact with fluids, probably rich in aloe oils, aloe, and myrrh. So this has been identified under the microscope. This is postmortem blood. So this is a paper. This is not related to Shroud research, but this came out in 2017. It's called Blood or Spores, a cautionary note on interpreting cellular debris on human skeletal remains. So they take red blood cells, they pull them out and kind of follow them over a few days. So they kind of stick together and start to bleb out a little bit. But most importantly, what they show in this study, they show images of various spores from various organisms that have a similar shape and similar size as red blood cells. And this is material that's relatively fresh. And then I think one of the coolest things they did, they took 60 photographs, and in these photographs were genuine red blood cells, and then also some of these spores from various organisms. They mixed the photographs up, and in a blind fashion, they gave them to 6 pathologists who routinely look at this kind of stuff, and 6 natural scientists who look at this kind of stuff routinely, and asked them to score it. Well, is this a red blood cell or not? So about 12%, they were unsure as to if it's one or the other. And then they actually got more wrong than they did right. It was about even in both groups. It was about 50-50. So these are professional practicing scientists who themselves do this routinely. Again, this is relatively fresh material. They have trouble identifying, well, is it really a red blood cell or not? How would you tell if you had something like this, if it was truly a red blood cell? Well, an immunologist is going to probably look, well, antibodies could do this. If you used an antibody specific for red blood cell proteins, red blood cell markers that are only found in red blood cells, you could do this kind of microscopic examination and it would light those up. So you could see that a shape, and then it would also be coded. It would show up as kind of dots with this particular approach. But that would would give you confidence that you're really looking at a red blood cell. So I think there's questions as to whether that's even a red blood cell or not. But, trying to classify it premortem or postmortem, I just don't think scientifically you can do that. And here's the kind of not counterpart, but additional thing they do. This is looking at photographs of the shroud and then based on color, identifying this as premortem or postmortem blood. So traces indicated by the blue spot in the figure appear to be postmortem blood leakage, which probably occurred due to movement of the corpse during transport in the tomb. And the red spots that are scattered throughout, those seem to be premortem blood, which probably occurred when Jesus was steel nailed to the cross, or was transposed, dissolved in the human environment of the sepulcher. So this is based on color. And what you don't see in this study is, well, if you take genuine premortem blood or postmortem blood that's aged, how do those sort out with this kind of colorimetric analysis? I can tell you from some of the blood work I've done, when you put it out and as it dries, you'll get some variation in blood that's guaranteed to be premortem. So I don't think you can really make these kind of distinctions. Again, there are no currently established tests that allow premortem and postmortem blood to be identified in a cloth 700 to 2,000 years old. It's not like you can take the fibers and test them and, well, this shows up to be premortem and this shows up to be a postmortem, which I think is kind of inferred by the way this is presented. And a lot of this is based on position. Well, we know that the side wound, we know that according to the Gospels, Christ was speared in the side after he had passed away. So this has to be postmortem blood if it's coming from that. Scientifically, objectively, you have to separate the bloodstains away from the context of the image. So these are the 3 maxims I think you have to follow when you're presenting or discussing these kind of scientific facts. Objective and unbiased presentation of information is essential, even when the results may be inconclusive. A desire to build a case for shroud authenticity, it should not necessitate the need for embellishment of scientific results. Exaggerations about the thoroughness and validity of shroud blood studies, This only serves to erode any previous credibility that may have been legitimately established. So I don't think scientifically you can't really say that, well, these have been tested and this is found to be premortem and postmortem blood. I don't think that claim really has any traction. And I've asked the same kind of thing. I've asked this question to people that work with blood and those kind of situations, and they said, no, you can't do that with something like this of that age and exposed to those kind of conditions. And you have to do it independently from the image if you're going to be scientific and objective about it.
Let's say the Pope calls you up and he says, Dr. Kearse, I want you to test the shroud. How would you go about doing that?
I would say, I would say, wow, is this really the Pope or is this my brother playing another trick on me? That's probably what I would say. I would say what you want to do is you want to get the best scientists that do this routinely every single day, and that's their specialization. You want to get outside scientists that have nothing to do with the shroud to test them. That would be my own personal recommendation. Yeah. I would probably send him to, I think the NIH has the best group of scientists in the world. I'm kind of biased of that, having worked there, but kind of biased that way. But I would say get outside scientists involved in this that are going to be objective and that do this every single day. And that's their specialty. That's what I would say. And also think about real carefully what you want to test. So if you want to go back and try to verify some of these things, is it human? Is it blood? Is it human, male, AB? I would forget about the male, trying to verify that. The human designation, I think it's important to validate that scientifically just because it's been talked about so much. But as long as you say it correctly, like is on the center of the Turin Centro, their statement is very correct. It has not been determined. It's still to be determined. So either say it correctly or have scientific evidence to back it up. So I think very carefully decide what it is that you want to test, because of course there's only so much limited material, and some of these tests by nature, they are destructive, which means it'll destroy the sample. So you have to be very careful, very reverent about that. You have to take it into consideration. But if the Pope called me, I would recommend outside scientists with nothing to do with the shroud. I think you're going to get the best result. And people that's their specialty. That's what they do every single day.
And could they find out the blood type too?
You could do the blood type. You would have to, so it depends. The best way, the foolproof, is to go for a DNA-based method. Whether the DNA is in good enough shape to do that, that's going to be an iffy thing. But just to point out again, even if they found a blood type, to try to connect it to various objects, you cannot use a blood type to do that to really be confident that it shares the same origin. You would have to use markers that are unique among people. Like the transplantation antigens would do that, or certain, as they typically do in DNA profiling, some of those markers would do that. Blood type is not going to get you there. So which is kind of what they're trying to do. I think a lot of people with the AB is to show that it's shared. You have to really nail that. You would have to go for something that's unique and polymorphic. The HLA, you can do if the DNA is not in such great shape. The HLA, the transplantation antigens, you could do that at the DNA or the protein level. Proteins tend to stick around a bit longer, so maybe that's more of a possibility there. If that's the goal, to try to make that connection. But you could tell that. You could definitely tell whether it was a human or not. That's a very straightforward kind of test with the antibodies that are now available. Absolutely, yeah.
And is there like a certain blood stain you would want to test?
So if I were kind of doing this, what I would do is go to the reverse side because they soak through. So you're not going to disfigure anything on the front. I would go through the reverse side. I would do just like Bima Bologna. I would come in with forceps and take a thread. He used forceps to remove threads, and there was no apparent disfigurement, or you couldn't tell any before and after from him taking the samples. I think it was a very, very smart thing to do. But I would go on the reverse side. You could even maybe spread it out a little bit and get in between. I would go for that part of it. Yeah.
I don't know if you want to talk about this, but is there any new stuff you're working on test-wise?
I am. So I'm doing some more of the why the blood is red. That's in progress. Some other stuff don't really want to talk about too much. And then another thing is the Maillard reaction, that was the idea of Ray Rogers. I'm still continuing that. I think you might have asked me last time about if hair transferred or not. So I hadn't done that the last time. It was just skin proteins. Hair does transfer. I can confirm that it transfers like the skin proteins. The proteins from hair transfer, protein breakdown products transfer. So I knew they reacted the last time that I believe I talked with you, but it does transfer. And I think the Maillard reaction is something that a lot of times it's dismissed in a lot of these theories because, well, there's not really contact, with parts of the body. So this can't really be true. But the fact that 85 to 90% of the body or so does make contact, that's not really taken into account. And when you have, Ray Rogers had a diffusion type mechanism, which things don't have to touch for that to work. So I think when you put the two together, I think it's a possibility. And then another thing that often is easily dismissed as well, if the shroud was wrapped really tightly, then you would see a lot of this distortion when you open the cloth. So it couldn't be just some kind of contact transfer. I personally don't think the cloth was wrapped very tightly around the body because they were working against the clock. They were in a hurry because the sun was going down. They knew they were coming back the next day to finish off the preparations. That's why they came back Sunday morning, why the women came back to finish the preparation. So I think kind of my own idea is they just sort of draped the cloth over his body. I don't think they wrapped it up and things like that. Why would you do that if you knew that you were coming back the next day to finish things off? So I think if you kind of have a sort of a draping configuration, that's going to cut down on that type of distortion that a contact would provide. And I think it could be a combination of things. So you could have a radiation type idea, but I don't think you can overlook the biologicals that would be in place for a Maillard reaction. You need two things for it to happen, A and B. And B is the protein degradation products. They're there on a body and they will transfer to a cloth. And on the cloth, you need the other component, which it's called a reducing sugar, which Ray Rogers chemically tested shroud fibers, and he said that was present. So it's going to happen. Whether it's the main cause of the image or you have something else, don't know. I mean, I'm pretty much open to any kind of possibility for it. I think a lot more work needs to be done on the image. We know that double bonds cause it, but exactly what particular form, is that cellulose or something else, that's not known. So so when you take single bonds between carbons, it's colorless. And when you make alternating double bonds, that's what gives you color, called a chromophore. So that's what the image, when we see the image, it's alternating double bonds that are present. So that could be the cellulose itself, where those double bonds are, or it could be something on top of the cloth, which is what Ray Rogers advocated. And so Ray Rogers, one of the interesting properties, it's only on that top, 200 to 600 top portion of the cloth, super, super thin, superficial layers where we find the image. Well, Ray Rogers took this material that he thought was coated with the cloth. It's a type of detergent material. He mixed it with dye, added it to the cloth, and through evaporation, it goes up and resides on that very, very top superficial part. And when he mixed the two things together, he got a color similar to what you find on the shroud. When he looked at it under the microscope, he looked at more fibers of the shroud than anybody, I believe, as far as looking carefully at the products and numbers of fibers. He found it was identical. These are his words, identical to what's observed on the shroud. So I think you can't necessarily dismiss that you could have a Maillard reaction as part of it. And one experiment that they did when they were looking at the shroud fibers, so they took those fibers that were colored, which had these alternating double bonds, they treated them with a chemical that would take it back to just single bonds. So when they did that, it went colorless. So it went from having color back to colorless. When Ray Rogers saw the results of those experiments, he was adamant that cellulose is not involved. He thought that indicated that was something on the top. I've talked to various chemists. Well, what does this say? When you do this kind of reaction, does that mean that cellulose is not involved? And they said, perhaps, maybe it's an accessibility thing, that if the double bonds were in the cellulose compartment, it would not flip back when you do that type of experiment. Rogers was a chemist. He was no dummy. Chemistry is what his specialty was. So I think that kind of hypothesis should be considered along with the others. I mean, I don't have any problem with any of it. I just, you want to see data that helps to back it up. And again, it could be some kind of combination of it, when they do these sort of treatments to mimic the coloration of the fibers with lasers and things like that, I think they ought to show that same experiment that it flips it back to colorless. I think that's an important demonstration. I really haven't seen that done yet. There are a lot of different routes that could lead you to that coloration on the fibers. You could do it with, I think, a mild type of heating. Now, the shroud's not a hot temperature scorch. That's been ruled out, but I don't think low temperature situations have been. You could also create it with acid, similar coloration, and you have that problem of restricting it to the upper part, but I think he could work that kind of thing out. And also the Maillard reaction of Rogers, a member of STIRP, I don't think that's really been given as much voice as it deserves to be looked at. And maybe that's not the mechanism, but I think it deserves looking into. And it's carbohydrate based. So carbohydrates are pretty much why I got into science from as a kid. That's sort of what hooked me, because that's involved in organ transplantation. Because blood groups were the first transplants before organs were transplanted. And that's all carbohydrate difference is what the immune system is recognizing carbohydrates. So I've had a long-standing interest in carbohydrates. So the Maillard reaction involves carbohydrates. I did my first postdoc. I got my degree in immunology, but my first postdoc at Hopkins was actually in carbohydrate biochemistry because I'm just interested in that. I wanted to pick up those methods, take them back and apply them to immunology. So the Maillard reaction with the carbohydrate part, it was just sort of naturally attractive to me, which is why I kind of was intrigued by it. I'm kind of wide open to anything. The more data you have, the better.
Did you have an opinion on the spices that were used or not used?
Yeah, so there's conflicting information on that. STIRP examined for that. They have pretty unique structures. They used some pretty sensitive tests. Rogers says there was not any a positive confirmation for aloe or myrrh. There have been two reports that it's present. One is by Natowski. It's a microscopic examination. Well, this is kind of shaped like, I don't know if aloe or myrrh or both, I can't remember, but it's shaped like that. So it's probably aloe or myrrh, but there's no real confirmation because I think a lot of things could kind of be shaped like that, sort of like the red blood cell situation we talked about. The other person that's looked at that is Byman Poloni. He did antibody approach. His study, I felt like it left the last control part out. He didn't really provide evidence that his antibody was truly specific for aloe and truly specific for myrrh. I think if he had done that, it would have either confirmed it or it would have showed a different thing. So I don't think there's any solid scientific evidence that those exist on the cloth. Stirp looked and said no straight up. Just those other two kind of coincidental observations are all that's out there. So I don't really know. And even what form, that might have been in. Some think it was dissolved in oil and perhaps it was going to be put on the body. Others have described it as like solid blocks in bags that may have been placed around the body. So I don't really know about that. So I guess my opinion would be, I'm not sure. I don't think there's really been any evidence to show that they were going to do it. But I don't know if they really got around to it. And maybe they did, but I don't really know.
When they cleaned the shroud and they put it in vials, you wouldn't be able to test that for anything, would you?
So that's more kind of a carbon-14 type tests would be the ones I think that those are most used for. I mean, they've used some for DNA analysis just to show in general what types of DNA were present on there from people touching it or certain kind of plants or things like that. But as far as a bloodstain study, I think you'd want to go to some brand new material, use forceps and take it out. That way it's definitive. There's no question about it. I don't think it's one of those things you want to repeat, the fifth time it was tested. I mean, you want to just go for it. Just go for it and nail it straight forward. So it would be the vials not really useful for that part. At C14, the char, I think people really would like to try that and try to sort out exactly what's going on with the carbon-14 dating.
Did you see that? It was pretty recent, an article that came out, and I believe how do you pronounce his name is like Nicole. He was a bishop in like 1370.
Yeah.
They're claiming it's like the earliest writing of the shroud.
Yeah. So I'll say to that, history is really not my thing. So I don't know. I did kind of glance over some of the things that were said, but just like super quickly. History is not my strong suit by a long shot.
I gotcha. Oh, how's retirement been?
Great, man. It's fantastic. Oh, it's fantastic. Yeah. Thanks for asking. It's great. I love the flexibility, of it. I work as a adjunct immunology college instructor, but just one class. That's it. And then it's fantastic having time to do a lot of things and around the house and just experiments. It's great. But thanks. It's been terrific. I miss being at school. I miss the kids and stuff, but it's great. I still see a lot of them around town and whatnot.
You still doing photography too?
I do. I still am. I'd like to do that more. I thought when I retired, I'd be going to the zoo a lot more and doing that, but I've just been kind of so busy with a lot of stuff that I haven't. So it's kind of gets pushed to the back burner, but absolutely still doing that. The Zoo Knoxville here, they're going to have a baby gorilla in a few months. So I will definitely be amped up and ramped up on that. I'd be a frequent visitor.
Is that your favorite thing to take pictures of is animals?
It is absolutely 100%. Yeah. Apes. I absolutely love apes and pileated woodpeckers up in the Smokies. That to me, the woodpecker is kind of more exciting than a bear. For me, it's always fun to get bear pictures, especially cubs. But I absolutely love those woodpeckers. I just have forever for a long time.
Are those uncommon or?
They're pretty common up in the Smokies. There's some people even around Knoxville, they'll have them, depending on where you live, they'll have them in the backyard and things like that. But pretty common up in the Smokies. A lot of times I won't see one, but I almost every time hear them. You can have a really distinct call, you can hear them. But there's a couple of places, particularly around Cates Cove, I usually hit. That's kind of a hot spot for the wood. They seem to travel within that part. So it's one of my favorite things to do is go up there and just chase around woodpeckers all day. It's great. They're just, they're huge. They've got that big red crest, and they just absolutely hammer that wood. And then it's cool. Probably my favorite bird.
Well, all right. We covered a lot.
Yeah, thanks very much. Thanks for having me on. Really
Thank
appreciate
you
it.
for coming on.